Okay, thank you very much. So, well, let me tell you that being a physicist by training, in particular theoretical physicist, I would have never imagined that I would be standing here today. So I feel incredibly honored actually to receive this award, especially coming from SDB, a society that I love. It's full of great colleagues and friends. I also feel very humble because, as you know, this award honors Elizabeth Hay, who was one of the pioneers in the field and a great developmental biologist. And I'm also feeling very lucky because there are many other young scientists, including many probably in this audience, actually, who deserve this award as well. So beyond the individual award, I wanna think that this award recognizes actually all those physicists, engineers, computer scientists, mathematicians, and others that are like me, that come from a different discipline, but we share with all of you this love for understanding how embryos develop. And in this sense, actually, I think that the society awarding this to someone with an unusual profile like me speaks volumes of how welcoming and how open-minded this community is. So for that, thank you very much. Okay, so today I'm going to be telling you about our most recent efforts to try to understand how embryonic tissues are physically shaped in 3 dimensions. So in my group, let me see if this works. Oh, I'm trying to go too fast. So in my group, we're interested in morphogenesis, like probably 90% of the audience here. And in particular, we're trying to understand how functional structures, like tissues and organs, are physically, or if you want, mechanically sculpted during embryogenesis. Now, this is a very complex problem, as you all know, and it has many different aspects to it, but for the last 50 to 70 years, it's mostly been studied from a genetic and molecular perspectives, and a great example as the previous talk that we've heard. But if you look at and by the way, the progress has been spectacular. I know that you guys, many people here work on the molecular aspects, and there are many other things to learn, but if you look back, actually the progress is really spectacular. Now, if you look at the embryos that I'm showing you on the left, though, there is another aspect of this problem that directly pops out, and these are the physical and the mechanical aspects of the problem, right? You need to understand actually how all these movements are coordinated and how these movements are able to shape these structures. Now, this was acknowledged already a century ago by Darcy Thompson, but it's been only in the last 15 to 20 years that really quantitative approaches have been put forward to try to understand this problem. Now, however, if you compare our understanding of the physical or mechanical aspects of the problem compared to our understanding of the molecular aspects of development, we are really in prehistory. We really know very, very little. And the reason for that is that, well, one of the reasons probably for that only, is that we have a lot of techniques to interrogate the embryos from a molecular perspective, a lot of them. But it's been extraordinarily challenging to measure mechanics during development, in vivo and in situ. So in this talk, I'm going to tell you about the efforts in my group to try to overcome this problem. And in particular, I'll tell you about the new techniques that we've developed to measure mechanics directly in situ, in vivo, within developing embryos. And not only that, you essentially can use these techniques in any 3D multicellular system, organoids, 3D cell culture, anything that you want. And then I'll show you that with these techniques, we can actually try to answer how embryos are physically shaped in 3 dimensions, and I'm going to tell you about the mechanism, the physical mechanism of vertebrate body elongation. So let me start by telling you a little bit of how different species elongate the anterior posterior body axis. How do they make the head to tail body axis? So if you look early in development, and by early I mean gastrulation and a little bit after gastrulation, there is a consensus in the field that the mechanism is convergent extension. And this was first reported by Ray Keller and co-workers back in the 70s, 80s in Xenopus, and then it was discovered in germ band extension in Drosophila and in zebrafish and in many other organisms. And the reason why it's a very interesting mechanism is because it's very simple. So it's very powerful as a mechanism. And it works in the following way. Essentially, in the tissue, there is a mechanical anisotropy that is created. There are forces along the mediolateral direction of the tissue, and these forces induce polarized cell intercalation along the mediolateral direction. And because of mass conservation, if you intercalate along the mediolateral direction, you're forcing the tissue to extend along the anterior-posterior direction. And the flows that you generate in the tissue, although I actually mathematically draw this once, actually, but it's not a real flow in the tissue. But essentially, you generate, by doing this, you generate these flows in the tissue. And you can see it sketched here in the original papers by Ray Keller. And also, you can see here the pattern of the, well, I don't know, probably you cannot see it, but it's there, actually, exactly this pattern in zebrafish. However, if you now look at posterior body elongation in vertebrates at the later stages, now you go after somatogenesis has started, you see something that is a little bit weird from a convergent extension point of view. You see a gradient of anteroposterior cellular movement. So you see that the cells are moving posteriorly and a lot at the posterior end of the body, but actually they get kind of arrested. They barely move when you get actually to the anterior tissues, just before the somites, okay? And we were very surprised about that when we saw it, because, I mean, actually this was reported by the group of Olivier Pourquier in chick and Scott Hawley in zebrafish, and we were very surprised because this cannot be explained with convergent extension. So we decided to look a little bit more onto in these flows and these cellular movements, and here I'm showing you a time lapse. Actually, you're seeing all the cells in the embryo, and now we can track all the cells, and I'm going to show you both the cellular movements, and I'm sketching on top of that the flows in the tissue. And the first thing that you see is that, yes, the cells are moving more at the posterior end of the body, and they are kind of arrested, or like they move far less in this anterior side, exactly like Scott Hawley reported for zebrafish. But you see these flows, you see that the cells are moving posteriorly here, they are arrested here, and there are two counter-rotating vortices in the tissue. And this was very surprising to us. I mean, convergent extension cannot explain this. So we decided to look a little bit more at the origin of these flows and try to understand actually what was the mechanism of elongation, the physical mechanism of elongation. And if there is one thing that we know in physics, is that if you see any movement in an embryo or any movement in a cell, it's completely controlled at the physical level by the forces, by the mechanics in the tissue, not necessarily by only the forces. So what we wanted to understand, what we're going to do actually is we're going to try to understand what is the underlying tissue mechanics that controls the movements that I showed you, the flows that I showed you in the structure, and the process of elongation itself, of posterior elongation. And we do that in zebrafish, and what I'm showing you here is a lateral view of the zebrafish embryo developing. You can see here the head and the tail here extending, and what we do is I don't know if I can stop here, this movie. Let me try it again. Okay, but essentially, we study the process at about when the somites have started to form, when there are about 10 somites in the embryo. And if you take a cross-section of the ventral tissues here, and you were to look from the dorsal side, you would see something like what I'm sketching here. You would see this is the posterior end of the body, okay, and it's elongating posteriorly. And here what you have is a region where you have mesodermal progenitors, and these mesodermal progenitors are differentiating into mesodermal cells to build the presomatic mesoderm. And then at the anterior end of the presomatic mesoderm, you have the somites. Okay, you have actually the embryos being segmented actually at the end of the presomatic mesoderm. Okay, so what we said is, well, since there is a gradient in the movement of the cells, very likely there is a gradient in the forces in the tissue. So this is, I don't know if you're familiar with this, but in order to shape tissues, what the idea in the field is that you have forces, spatial and temporal changes in the forces that guide the formation of the structures, right? So we said, okay, let's measure the forces in the tissue. But, well, we were lucky enough that we had developed a technique to do that, okay? So to measure forces in living tissues. And the technique is very simple. We use oil droplets as force transducers. So imagine you have one of these droplets, and by the way, these droplets are not as simple as taking the oil from the kitchen and just putting it into the embryo. That doesn't work, huh? So they're a little more fancy. But they are all droplets, and the idea is we put these droplets in between the cells in the tissue, and the forces around the droplet, actually the forces in the tissue around the droplet, deform the droplet, and by just looking at the deformations of the droplet, if you have calibrated these droplets, you can know what are the stresses that the tissue is doing on the droplets. And you can have these droplets in the tissue, and they are tracked by morphogenetic flows, and you can actually, at any time point, you can reconstruct in 3D this droplet and obtain what are the forces that the cells and the tissue is applying on the droplet, right? So we decided actually to go use this technique and measure the forces along the anterior-posterior axis of zebrafish, and what we find is the following. This was very surprising. We found actually that there was no gradient in forces. It was uniform in the tissue. So the magnitude of the forces that the cells are applying is uniform in the tissue. And this was very surprising for this idea that we thought actually that, well, if there's more movement in the posterior end, there's going to be more forces. The cells are going to be applying more forces in the tissue to induce more movement. So we were very surprised, and we wanted to learn a little bit more about the characteristics of these forces. So what we decided to do is to look at the persistence of these forces. And the reason for that is that if you want to build a structure, any structure you want to build, in particular you want to shape a tissue, the forces, if it's the forces that are shaping them, that is shaping the tissue, they need to persist over developmental timescales. Because if they don't persist over developmental timescales, it's impossible for them to build a structure. So we measure the persistence time of the forces, and what we find is that they persist for 1 minute. Just 1 minute. So this was very surprising to us, actually. Essentially what is happening is that in the tissue, the cells are kind of pinching the tissue randomly, in all directions. But there's no coherence in the forces. It's not that the forces are collectively shaping the tissue. So we were very surprised. These forces could not shape, were not explaining the observed movements and could not drive axis elongation. So we were very surprised by this. So what are we missing? We clearly were missing something. And at the end of the day, what we're trying to do is we're trying to understand how a structure is built, right? So it's very informative to look at how we humans build the structures. And if you look at how we build the structures, how we make functional objects like the chair or whatever actually you want, you realize that we have several methods to do that. We can actually tune, you can build an object by tuning where and when you put material to the structure. And that's, for instance, like 3D printing. All of you have probably tried that. Actually, in 3D printing, essentially the machine goes and puts material here and there to build the structure, right? You can tune the forces, yes. You can actually apply forces here and there in a material to shape a structure, and that's like clay molding. It's exactly the same. But you can also tune the material properties. What it means is, like, for instance, focus on glass molding. You can actually warm up a material, like turn it into a fluid and shape the structure, and then actually rigidify it again by cooling the material, right? Like in glass molding or glass blowing. So, well, it turns out that it cannot be, that the body axis cannot elongate through addition of material, because the group of Scott Hawley actually showed already a long time ago that he did actually some cell cycle mutants, and he showed actually that cell proliferation was not elongating the body axis. So you can inhibit cell proliferation, the body elongates in the same way, it's just that the cells are a little bit bigger. So it cannot be cell proliferation. Well, it cannot be the forces, because I just told you that the forces actually in the structure are not actually elongating the body axis. So what about the material properties? So we wanted to know if it was the material properties. The problem is that there were no techniques to measure material properties in vivo and in situ, and in particular spatially inside the embryo. So we had to develop a technique. So I'm sure that all of you know how to measure material properties, even if maybe you have not wondered that. But in order to know if something actually is soft or hard or whatever actually it is, what you do is you poke on it. You actually apply a force, and you look at the response of the material. This is what you do, right? So this is what you need to do in an embryo now. So what we do is we took the droplets that we had, and we turned them magnetic. So we use biocompatible ferrofluids to do that. And the idea is that if you now apply a magnetic field on these droplets, you deform them. You can deform them, and you can apply forces to the surrounding material, right? And what happens is that when you apply the magnetic field, the droplet deforms, but then when you turn off the magnetic field, because this is a droplet, this has a surface tension, right? Like, imagine a balloon, right? You pull on the balloon, and then when you let it go, it comes back, because it has a tension. So the idea is now, you're going to put this droplet inside a tissue, and then we're going to apply a magnetic field, and we're going to poke on the cells around, and we're going to see how the tissue responds. And this is exactly what I'm showing you here. This is exactly what I'm showing you here. You have a droplet, we apply magnetic, we turn on and off a magnetic field, and you can see that we can poke the tissue, right? So we took these droplets and we put them in the tissues during body axis elongation, and then actually we deformed them. And here actually we were playing a little bit with it actually, and we were deforming in different directions. You can play with the direction as well. But so what is the information that we get out of this? So the experiment that you do is as follows. You actually put the droplet in, it stays, by the way, you don't put it in and you start the experiment. You need to let the droplet inside, stay inside the tissue for some time so that the hole that you did during the injection heals and all that. But essentially when the droplet has been embedded in the tissue, then you turn on the magnetic field and you can see that the droplet deforms. This is the strain that you apply in the tissue, but you can think of that as the droplet deformation. So the droplet deforms more and more and more, and then what we do is we turn off the magnetic field. And as I said, there is a tension in the droplet, right? This interfacial tension. So the droplet is pulling back to go back to the spherical state. And what we observe is something a little bit weird. We observed that the droplet was not going back to the sphere. There was a reminiscent, if you want, a residual deformation on the droplet. So something needed to be holding this droplet. And this something is called, in physics, a yield stress. Now, many of you may not know what a yield stress is, so let me explain you what it is. So let me show you first materials that I'm sure you're going to be familiar with that do have a yield stress. So all of these materials have yield stress, and actually many of the materials that you see in your life actually have yield stress. So these are materials that if you don't apply any force to them, or very small forces, they behave as solids, elastic solids, okay? But if you apply a force that is large enough, in particular above a threshold that we call the yield stress, the material starts to yield and it flows like a fluid. So these are materials that can switch between fluid and solid states depending on the stresses in the tissue, or in the structure in this case. So from a developmental biology perspective, this is very powerful for one reason, because during development, the tissues need to behave as fluids. You need to shape the tissue. It needs to behave as a fluid. But you need also to maintain mechanical integrity. Once you've shaped a tissue, you don't have the eyes in zebrafish when you've shaped the eye actually like dripping like a fluid, right? You need to maintain somehow mechanical integrity. You need the tissues to behave as solids as well. So this could be a potential mechanism. In other words, the embryo could, or the cells could play a little bit with this mechanism to tune whether it's a fluid or a solid. And we wanted to know whether this was the case, so we measured the yield stress along the anterior-posterior body axis. And what we found was that the yield stress was indeed graded along the anterior-posterior body axis, and it was minimal at the posterior end of the body, and it increases away from it, and it's maximal in the region, in the anterior PSM, in the region closer to the somites. So what we wanted to know when we saw that was, well, what is causing this yield stress, and why is it graded? What's causing this gradient? So if you look at the tissue, like here I'm showing you a confocal section through these tissues, and you're seeing actually these are the membranes being labeled here. So what, you look at the tissue and it looks like a foam. You just look at it and it looks like a foam. And by the way, I need to mention one thing. There is no matrix between the cells here in the tissue. So this actually was showed by Scott Hawley as well. If you look at fibronactin matrix, there is no matrix between the cells. All the matrix is surrounding these tissues. There's a sheath of matrix surrounding the tissue, but it's nothing in between the cells. So, and if you look at it, it looks like a foam. So it turns out that we know in physics that foams do have a yield stress. And I'm showing you here one that probably is one of your favorites. So, and this foam here actually does have a yield stress. And the yield stress actually in foams depends strongly on one parameter. It's the volume fraction of fluid in between the bubbles. So if you look at this foam down here, well, I mean, the bubbles inside the beer here, actually that would be a large volume fraction of fluid, and there is no yield stress. This behaves as a fluid. You all know this, right? It behaves as a fluid. But actually what happens is that when the bubbles start reaching the top here and they pack up in this region, what happens is that now they start becoming constrained by the neighbors, by the other bubbles. This is called, and this point here is called a jamming transition. What happens is that the bubbles jam, and their movements start being very constrained. And at that moment, the foam starts developing a yield stress. And if you go up in the froth, now you go into the top of the froth of the beer here, now what happens is that you have very little fluid between the bubbles, and you are down here, so you have a very large yield stress. Think about this for a second. What you have here is bubbles that are made of air. They have just a little thin layer of fluid around them, but collectively they behave as solids. They behave as a solid. As a soft solid, but as a solid. So, so this is actually the physics of foams, and we actually thought, well, maybe the tissue behaves in the same way, and this is why it has a yield stress. So we wanted to know if that was the case, and for that, we wanted to measure what was the volume fraction of the spaces between the cells. So how did we do that? Actually, so we decided to measure it by injecting fluorescent dextran into the tissue, and the fluorescent dextran would reveal the spaces between the cells. And if you measure the volume fraction of the spaces between the cells, what you find is the following. you see that in the region where the yield stress is maximal, the amount of spaces between cells is minimal, and vice versa. Where the yield stress is minimal, the volume fraction is maximal, or, well, I mean, it's not exactly, but it's the case. But this was only a correlation, right? So what we wanted to do was to do a perturbation of the spaces between the cells and measure the yield stress. You want to perturb and see what comes out, right? So in order to perturb the spaces, what we did is use n-cadherin mutants. We used the parachute mutants. Because we thought that if you actually have less n-cadherin, well, maybe the cells have more spaces between them. And indeed, if you measure the spaces between the cells in the n-cadherin mutants, what you see is that in the anterior PSM, you open a lot more spaces. But actually, surprisingly, in the mesodermal progenitor zone, in the tailbud region, the spaces don't change. You have exactly the same spaces between the cells. But this was great for us because it's having a built-in control into the system, because what you expect, if this picture of the foam is true, what you expect is that the yield stress will not change in the posterior end of the body, and it will change maximally on the anterior end. And when you measure the yield stress in the n-cadherin mutants, this is exactly what you see. Exactly what you see. And not only that, what you see is now that the yield stress, in the n-cadherin mutants, the yield stress does not show any anterior-posterior gradient. So somehow the gradient of the yield stress, the gradient of mechanical integrity in the tissue is controlled. Well, no, I shouldn't say it's controlled. It's dependent on n-cadherin. So what we learned from these experiments is that the tissue behaves physically like a foam, like a 3D disorder foam, and that the gradient in the yield stress is controlled by this gradient in the spaces between the cells, which is n-cadherin independent. But I haven't told you whether the tissue is in a fluid state or in a solid state. And the reason is because in a yield stress material, you need to know what are the forces in the structure. You need to know whether they are above the yield stress or below the yield stress, as I said before. But luckily for us, we had measured the forces. So what we did actually is compare them. And what you find when you compare the forces and the yield stress is that at the posterior end of the body, in the mesodermal progenitor zone, the stresses are consistently larger than the yield stress. But when you go actually to the presymmetric mesoderm, the forces actually in the presymmetric mesoderm start becoming smaller than the yield stress. So in other words, by just doing mechanical experiments, we could tell that the tissues were transiting from a fluid state at the posterior end to a solid state in the PSM, actually. So we were very excited about that there was a fluid to solid transition, and that was controlled by jamming, actually, like jamming in foams. However, there is something that doesn't fit, from a physics perspective, there's something that doesn't fit here. Because in order to maintain a foam in a fluid state, the only way that you have to do that is to induce rearrangements between the bubbles. That's the way actually you fluidize a foam in physics. And that actually normally is by applying a stress. So you actually take the foam, well, I mean, going back to the example of the beer, if you go and you blow on the beer, on the froth of the beer, you blow on it, you apply a force, and then actually becomes a fluid. You can actually irreversibly deform the foam. But what you're doing really is you're doing rearrangements of the bubbles inside the foam. And that's why it can flow. But there is nobody here externally applying a force actually on the tissue and inducing these deformations. However, this is not a normal foam. This is not like the beer. This is a living foam. And as a living foam, it has cells there that can apply actomyosin contraction. They can apply the forces that we were measuring here through actomyosin contractility at the cell-cell junctions, and therefore they can induce internally cell-cell rearrangements. So we decided to measure the frequency of rearrangements in the tissue, and when you measure the frequency of rearrangements, what you see is that at the posterior end of the body, in the region that is fluid, you indeed see way more rearrangements, actively induced rearrangements in the tissue. So what is happening is that, remember in the beginning that I told you that the forces in the tissue were kind of like random forces, that they were fluctuating everywhere the same, so they could not extend the body axis? Initially, we thought they were useless, these forces. Obviously, the cells would not be doing that, actually, unless it was useful in some way, actually. So they are like doing all these fluctuations, and these fluctuations are driving more rearrangements between the cells and the posterior end of the body because there are more spaces between the cells. It's easier to unjam the cells in that region. And therefore, these rearrangements between the cells are melting the posterior end of the body. And they're fluidizing maintaining it into a fluid state. So let me put this together. So essentially what we find is that the posterior tissues or the tail bud is maintained in a fluid-like state, and this enables the extending body axis to be remodeled during body axis elongation. And that is done because the cells have more spaces between the cells, so it's easier in that region to uncage them from the neighbors. They are less constrained by the neighbors. And also, you have these fluctuations, these force fluctuations actually that are driving these neighbor exchanges and fluidizing the posterior tissues. And as the cells progress into the PSM, what happens is that they close down the spaces between them, so it's much more difficult for them to rearrange, and these force fluctuations, they cannot actually induce these rearrangements, and the tissue becomes a solid. And then essentially what's happening is that this solid-like state of the tissue establishes the tissue architecture. It fixes, it makes it mechanical, it gives mechanical integrity actually to the tissue. And from a physics perspective, this mechanism is very similar to glass molding. It's nearly analogous to glass molding. It's just that obviously there is nobody externally with a blowtorch actually there, like changing the temperature or melting the posterior end of the body, but actually it's all the cells internally in the tissue that are doing that by themselves. And obviously I want to point out here that I just focused this talk on the physical mechanism of elongation, but actually obviously there are signaling molecules that are controlling this. There's a lot of cell behaviors controlling these fluid to solid transitions, and in my lab we are now studying that. We're studying the role of several signaling molecules and how this signaling is controlling actually cell behaviors to control all this physical level. But if you're interested in that, we can talk about it later. So this is okay, but I told you about the physical mechanism. I told you about the physical state of the tissue, but I didn't show you at any point that this can explain the morphogenetic flows in the tissue, and this can explain axis elongation. So the only thing that I told you is that you have a fluid, the tissue in a fluid state at the posterior end, and that then it becomes solid in the PSM. And another thing that I didn't tell you, but actually is happening, is that there are cells that are coming from dorsal tissues, and that these cells from the dorsal tissues get inserted in this fluid-like region in the mesodermal progenitor zone. Essentially, it's like adding material to that region constantly. So if you take only these two ingredients, the fact that there is a fluid to solid transition and the cells entering from the dorsal tissues, and you simulate this according to the physical laws that we know, what you see is the following. When there is a fluid to solid transition in the tissue, you get unidirectional axis elongation with large velocities at the tip. But if you don't put the fluid solid transition, you actually get a blob, a growing blob. And now you can say, well, I mean, this is not the only thing that you predict, actually. We can actually predict the morphogenetic flows in the structure. So if you look at the prediction for the morphogenetic flows, what we get is the following. We get large posterior velocities of the cells here. We get the cells are arrested in this region here. And we get two counter-rotating vortices in the tissue. And I just want to remind you of our measurements. We have two counter-rotating vortices exactly the same that we're observing here, and the posterior extension here. All these predictions just come out of this transition and the fact that you're adding cells from dorsal tissues, nothing else. So let me finish by thanking everyone who was involved in this work, and also I want to thank other people actually who helped me enormously during my career actually, and in particular I wouldn't be here today actually if it was not for them. So I want to start by thanking my lab, the former members and the present members. I've been extraordinarily lucky to work with an outstanding group of people, and it's all the credit goes to them. I mean, I didn't do any of this work. And in particular, I want to thank Friedhelm was the person, actually an experimental physicist, who developed the magnetic droplets techniques that I showed you, and he did that with the help of David and Zach. David is a physicist, and Zach an engineer. Alessandro, who was actually mentioned in Yanni's talk, was a graduate student with Yanni. Ale did all the mechanics measurements that I showed in Zerofish with the help of Hannah. Adam was the person who kept in check all our droplets to do the force measurements, and Elijah, our force master, is the person actually who measures all the forces in tissues. He is an engineer, he's a physicist, and Payam, a theoretical physicist, was the person actually who did analysis. I didn't show you this analysis of junctional fluctuations in the tissue, but he did all this analysis with the help of James. And he also did the simulations with the help of Emmett, who actually was an undergrad that ran nearly all the simulations. And finally, Georgina, who is a developmental biologist who's done all the analysis of morphogenetic flows. So I want to point out, actually, these are theoretical physicists, experimental physicists, engineers, and biologists all working together to do this work, actually. So it's really spectacular that they could pull it off, actually, together. And I also want to thank the rest of my group, because they were either directly or indirectly related to this work. They help in different ways. I want to thank my collaborators in other projects, Ophir Klein, David Kimmelman, who I'm sure that most of you know, Ellen Sletton, and Jerome Groh. I also want to thank my funding sources, actually, in particular, NICHD. Nearly all the work that I show here was funded by NICHD. And I want to take this opportunity also to thank the people that kind of shaped my career and that supported me along the way. Particularly my PhD advisors and my postdoc advisors. As you can see, I've been very lucky to have 3 PhD advisors and 3 postdoc advisors. I would recommend that to any trainee here. I actually have many advisors. And you may not know most of them, because they are physicists, applied mathematicians, and well, you may know Don Imber, actually, who is a developmental biologist and a bioengineer. But all these people are outstanding scientists. If you don't know them, I can guarantee you that. Outstanding scientists that helped me enormously along the way. And I want to thank them for that. And finally, I want to thank all the people involved in one event, one experience that I had in my life that completely changed my career, and actually my life as well, in many different ways, and that sparked my interest for developmental biology. And that was the embryology course at Woods Hole. I want to thank everyone who was involved. Actually, anyway, I don't have to mention this. No, I mean, I get a little bit emotional because I met my wife there, actually. So anyway, so I want to thank, actually, everyone who was involved, the students, the TAs, the professors, the directors, Lee and Ipam. They all helped me enormously, and well, thank you all for your attention.